I'm Rick Lifton, and I'm the president of Rockefeller University, and I'm absolutely delighted to welcome all of you to this morning's Women in Science Spring Breakfast program. If this is your first time visiting Rockefeller, I hope you'll become involved with the university through our Women in Science initiative, which was launched 21 years ago to encourage women in the business and philanthropy groups to embrace life sciences for its critical contributions to advancing human health and society. If you go back 110 years, 17, 18 years, a child born in 1900 had a life expectancy of 40 years. Today, a child born this year will have a life expectancy, if you're a female, of 82 years, if you're a man, of 77 years. And a child born this year will have 1 extra year of life expectancy than a child born 4 years before, who will have a life expectancy 1 year greater than a child born 4 years before that. These incredible advances have come from understanding the fundamental issues relating to how human biology works, and that leads to our understanding of health and disease. And this is what Rockefeller University is all about. Through the Women in Science initiative, the university provides fellowships to emerging scientists and supports critical resources for advancing science across many fronts. If you're interested in joining Women in Science mailing list, please speak to one of our staff members immediately following today's program. Before introducing today's speakers, I have a couple of quick announcements. The 21st Annual Women in Science Spring Lecture and Luncheon, an incredible highlight of the academic year on campus, will be held on Thursday, May 10th, and will feature Agata Smorgazuska. Agata is a remarkable physician scientist here at Rockefeller, and she's an alumna of the university's graduate program who returned to Rockefeller in 2009 to direct the Laboratory of Genome Maintenance, and I promise you this will be a real treat. Agata will give a presentation titled DNA Repair, the Power of Precision Medicine in Treating Cancer, and her talk will focus on how new discoveries are fueling the development of innovative cancer therapies, including some that precisely target an individual's tumor cells by exploiting the cell's own defects in DNA repair. You can find additional information about this event on the back of your program, and tickets are now available through the Women in Science website. And now it's my pleasure to introduce this morning's first speaker, Dr. Elaine Fuchs, who holds the university's Rebecca C. Lancefield Professorship, a chair that was created by supporters of Women in Science in 2002, which enabled Rockefeller to recruit Elaine to join our faculty. And I can only tell you what a transformational recruitment that was for the Rockefeller campus. Elaine is the head of the Robin Kemmers-Neustein Laboratory of Mammalian Cell Biology and Development. She is, without question, one of the world's foremost stem cell biologists, and her work has absolutely revolutionized our understanding of skin biology. She has done incredible work over the last 40 years, which has garnered her the most prestigious honors in science, including the Albany Medical Center Prize, the March of Dimes Prize, and perhaps the most prestigious honor, the National Medal of Science, which was awarded to her by President Obama, one of the highest accolades in American science. Elaine's research focuses on a deceptively simple question in development. How do you start with a single stem cell that can differentiate into many different cell types? And the demands for those different cell types varies locally and across the entire epidermis with time. How do you make just the right number of cells of the right type in the right place under particular demands? And she has really spearheaded this effort in her work. She started out characterizing the fundamental components of what's in the skin, the keratins, the elements that regulate the keratins. She used these tools to figure out a number of important skin diseases and really led this entire effort to understand skin, and one of the big advantages of studying skin is you can directly see it. And she has taken this to incredible advantage, particularly as we get down to stem cells. People have known that stem cells exist in specific niches and they have surround cells that are very important for maintaining the niche, but we've known very little about how these work. And the huge advantage that Elaine pointed out was that in the skin, you can actually see these, and they're well organized, and you can study them and manipulate them and understand how they work. And the work is absolutely extraordinary. Elaine also has identified one of the first cancer stem cells and has used this to understand one of the most important types of cancer, squamous cell carcinoma, which is a very common cancer that affects many different organs. So she's just done extraordinary science that has really transformed how we think about a field. In addition to her great basic science, Elaine and members of her laboratory have embraced the new entrepreneurial climate in biomedical research. Back when we were starting as graduate students, there was academic science and there was pharma. And the great biology coming out of academic science was very poorly connected to the pharmaceutical industry. That led to the development of the biotechnology industry, which helped translate. But we now realize that as these tools have gotten better and better, we can bring that further back into academia, where the people who really understand the biology can really run on these problems using tools that a generation ago you would only find in the pharmaceutical industry. So in particular, we're making use of the Robertson Therapeutic Development Fund to explore the broad biomedical potential of experimental findings. This was created 4 years ago by Rockefeller trustee emeritus Julian Robertson, and the fund awards grants that enable our scientists to advance their innovative research projects to a stage where they can attract outside industry partners. These are competitively awarded grants that support the projects that have the greatest likelihood to benefit patients. So joining Elaine this morning will be two scientists from her laboratory, Yejing Ge, who is a new research associate in her lab, and Ph.D. student Samantha Larsen. Both are recipients of Robertson Therapeutic Development Fund grants, and they are also recipients of Women in Science fellowships, generous and important funding that, thanks to many of you, helps to support the early scientific careers of women here at Rockefeller. So when Elaine was asked to give this talk, it was her idea to combine the story of her translational work with the story of great mentorship. She is an unparalleled talent in both arenas, and I am delighted to turn the podium over to Elaine. Please join me in welcoming Dr. Elaine Fuchs.
Okay. So let me start by just thanking Rick, and then also to thank the Women in Science organization for everything that you do for Rockefeller University, and in particular for women here at Rockefeller. And today I'd like to tell you about women in science, and it's an area that is very near and dear to my heart. One that I haven't spoken about for any of the various different breakfasts or lectures. And let's go back even less than 100 years, where if we're at a physics conference, there's likely to be one woman in the room, and that is if Marie Curie happened to attend that conference. And so historically, whether in physics, math, chemistry, or biology, the theme has often been the same. In Marie Curie's day, women were not encouraged to go to university, let alone to think about science as a profession. For the life sciences and for the medical profession, this has changed, but there are still hurdles. A major hurdle is that with regards to the qualifications all equal, the male is often deemed smarter and more accomplished. And this really permeates still publishing, scientific presentations, job interviews. In choosing speakers for scientific conferences, all too often, talented women are still overlooked. And finally, in a conference or a meeting setting, when a woman voices a good idea, all too often, a man will often reiterate it and receive the credit for it. And these aspects, while they've been changing, they haven't changed sufficiently, and we still have work to do. So just a couple weeks ago, there was a New York Times article, front page, where people were asked to draw a picture of a leader. And mostly they drew pictures of men. And in reading that article and looking for the explanation, one comes up with the stereotyped expectations rather than objective reality are driving that impression. And the solution is clear. We need more women in positions of leadership. So since Marie Curie's day, we now have a vast increase in the number of women coming into the biological sciences. 50% of our women students are women. Nearly 50% of our postdocs are women. But as you go up through the ranks to assistant professorship, tenure track positions, up through to full professor positions at major universities in the country, we're still finding that those numbers are dwindling further and further. And so what's the explanation for that? Well, often, too often, when men and women are not progressing up the ranks at an equal rate, it's often perceived as the woman's problem. Women are going off and having children. Women are spending too much time with their family. Women aren't motivated enough. There are many different explanations that have been tossed out there. But I'd like to just reflect a moment about whether the problem, and if the problem then maybe the solution as well, might be in terms of our mentoring rather than gender per se. And here I don't want to give the perception that everything, the entire solution is going to be with regards to mentoring, but I've been a head of a laboratory at major universities for a number of years, and there's a few things that I've learned over my experiences that I think might be useful in terms of moving to help correct or erase the gender disparity at the transition to faculty levels at top universities. And first of all, when the boss seeks everybody's opinion in the room, ideally before his or her own opinion is expressed, it sends a message. And that message is that everyone's opinion matters. And when everyone's opinion matters and everyone is brought out to raise their opinions, now those who are more quiet and resident, often women, tend to be confident now because they're asked to express their opinions. So everybody gains confidence when that happens. Importantly, the others in the room who are listening to those quiet and more reserved people express their opinions end up realizing that quiet and reserved doesn't equal a lack of knowledge or ideas. And so when that happens, then carrying this back to the laboratory, which is so important for science, interactiveness and idea sharing become part of the culture within the lab, and that moves everybody's science forward, men and women. Another very simple change is that when the boss welcomes challenge and criticism, and for us in science, criticism and challenging of our science is so important. It's important in order to be able to improve the scientific rigor within the laboratory. So we have to challenge and criticize each other's science to be able to really do the best science that we can. It's also important to prepare the lab members to be able to defend their science and their ideas in front of scientific conferences. And finally, one of the most important aspects here is that we must be clear that that criticism and the challenging are directed toward the science and not toward the individual. And with these very simple changes, which at least I've been practicing for now quite a few years, I think we can make a difference with regards to the gender disparity. And this gives you an indication of just how we can make a difference. These wonderful women here are all women who trained in my laboratory, starting at either the graduate student level or the postdoctoral level, and they've all moved up through the ranks, some of them now at the full professor level, those who were with me quite some years ago. I'll list for you that group, and these are not women who have transitioned to small colleges or teaching colleges or non-tenure track professions. They're not working in other people's laboratories. They're running their own laboratories. And they're at major universities throughout the world. They include Stanford, Harvard, Yale, Princeton, Columbia. And we have yet to hire someone at Rockefeller who trained in my laboratory, but hopefully that will happen soon. And when we look at the next big hurdle now, and I wanted to mention one last thing here, because effectively, the one thing I wanted to mention before is that I asterisked women who have had children. And many of those women who are now at major universities, full professors, had women and had children while they were in my laboratory. So it's not just a either-or situation. These are women who have had families and have gone up and been successful. I also wanted to mention a few examples of really great leaders that have emerged from that kind of a climate. Julia Segre, who is a former postdoctoral fellow in my laboratory, now heads the microbiome skin genome project at the National Institute of Health. Several years ago, she too received a medal from President Obama, this one, a public service award for taking the initiative to go out when there was an infectious, dangerous outbreak of a pathogen at a National Institute of Health hospital, and going out, sleuthing out, finding the pathogen, sequencing it, and eradicating it. Valerie Horsley was another example I wanted to mention, because she was a postdoctoral fellow in my laboratory, recipient of a Women in Science postdoctoral fellowship. She had 2 children while in my laboratory, is now an associate professor at Yale University, and has recently declared her interest now, and she is running for state senate in the state of Connecticut. So these are women who think big and do great science. So the next big hurdle, as I'm indicating to you here, is involving women scientists in translational science, in biotechnology, and in forming companies. And again, I draw us to an article a couple weeks ago in the New York Times where that discussed women's money problems. And here, pointing out that wealth is connected to power and that there's a real gender gap in both arenas. And when looking at the reasons underlying that, again, it's a cultural issue that women traditionally have been discouraged from the idea of going out there and making money. We haven't basically been educated to think that that is something that is worthy or important to do. And for those women who have, they've often viewed it not as a means to gain power, but rather as a means to benefit mankind. I don't want to argue too much with that latter aspect. I think it's a good thing about women and women who have attained positions of money and power. But I do want to point out that when we consider that there's a glass ceiling hear about women translating their ideas to positions where, to areas of clinical application, that's actually a good thing. And so we women should be exceptional at being able to translate what it is that we're doing, the basic science findings that we're making in the laboratory, and translating those to make it useful for clinical applications, for advancing medicine, for coming up with new and improved therapies for the treatment of various different diseases. So I want to give you a few vignettes of research that has been going on in my laboratory that takes the basic science and asks whether we can take those basic science findings and move those to clinics and to therapeutics. So as Rick pointed out, my laboratory for a number of years has focused on skin and skin biology. We've been interested in the basic biology of the skin, and we've studied, as Rick mentioned, the stem cells. These are not embryonic stem cells. Every tissue of our body, of our adult body, has stem cells, and they come in different flavors. They're important because whenever there's a dying cell within the tissue, the stem cells go out and repair it. Whenever there's a wound, the stem cells are mobilized to go in and repair the wound. And so normally they sit doing not much of anything in our tissues, but then in order to be able to do these various tasks, they basically become mobilized, they receive 911 signals from different regions of their tissue, and they go out and do their job. My laboratory has isolated and characterized the stem cells of the epidermis, of our hair follicles, of our sweat glands, and more recently, we've started to focus on the stem cells of melanocytes. And we've studied the basic biology of these stem cells. We've studied what are the factors that mobilize these stem cells? How do they perform these various different tasks? And that has led us now to a platform where we can start to address all sorts of interesting questions, some of them curiosity questions, and others medically important questions. So in the last year, my laboratory has addressed the simple question of why is it that we humans are unique in having sweat glands over our entire body. For any of you who have cats and dogs, on a hot day, their tongues are hanging out. Ours aren't. And the reason for that happens to be that we have sweat glands. We're much better at thermoregulation. So we can run marathons, your dog and cat cannot. We've worked out why it is that our hair is thin and gray as we age. We've been focusing on why our wounds heal more slowly as we age. And several of the papers that we published in the last year deal with these topics. Another area of interest is why does inflammation in inflammatory diseases, chronic inflammatory diseases such as psoriasis or atopic dermatitis, why does inflammation come and go, and when it comes back, it tends to come back in the same spots as before? Samantha Larsen, a graduate student in my laboratory who is going to be speaking after Yejing and after me, worked with Shruti Nayak, a postdoctoral fellow in my laboratory, both of them having received money from the Women in Science or fellowships from the Women in Science that honor them. The two of them figured out why that is, and they've got a very interesting story. Sam will tell you about it. And when you start to take a look at these kinds of questions of why do our wounds heal more slowly as we age, or why do we tend to get inflammation in the same places as before in these various chronic inflammatory diseases, now we're talking about science that, if we take it to the next step, takes it to the level of making a real difference in terms of clinics and therapeutics. And then finally, our focus has been on the stem cells that are giving rise to cancer. And what we've learned is that genetic mutations in tissue stem cells are at the roots of cancers. My laboratory studies squamous cell carcinomas. These are very prevalent cancers. They affect the skin, the esophagus, head and neck cancer, certain types of lung cancer, certain types of breast cancer, cervical cancer, a variety of different very life-threatening cancers. And yet we still have very few good therapeutics for the treatments of these cancers. We've identified the stem cells that are giving rise to these cancers, and we've come up with mechanisms where we can mark them in green, as you see on the left, and we can follow their behavior. And we've learned that these are the cells that become invasive and resistant to chemotherapy. Again, now recognizing the clear medical relevance to the findings, we just have to make it to that step. And then finally, in work that Yejing Ge will talk about right after I finish, we've looked in the last year at parallels between wound healing and cancer, and Yejing will talk about cancer as a wound that never heals. So how do we bridge then the gender gap in translating discoveries to medicine and biotechnology? And as Rick alluded to, we now have a mechanism of how to do that called the Robertson Fund that was started at Rockefeller several years ago. These are great opportunities, and both Yejing and Samantha successfully received Robertson funds to take their ideas to the next step. And yet, in order to be able to obtain Robertson funding, there needs to be strong preliminary evidence of clinical or pharmaceutical application and the prowess of how to see it through. And therein lies a major problem, because few women have applied for these grants, and therefore we're really not tapping in to the power and strength of all the great findings that are happening on campus. This would change if women received mentoring and training for entrepreneurship, and if their great ideas could be funded at an earlier stage than what Robertson typically funds. And therefore, I'm extremely pleased to announce that the Women in Science organization have now launched a Women's Entrepreneurship Fund at Rockefeller University. This fund will provide early stage innovation grants and entrepreneurial training to women at Rockefeller. This fund will be open to women scientists at every level, with emphasis on Ph.D. students, postdocs, and early career investigators. And this fund will equip a new generation of female scientists with the knowledge and skills necessary to develop breakthrough therapies and diagnostics, working with biotech, pharma, and venture capital communities. And so I think this really is going to make a difference, a huge difference, because think of all the great science that is being performed at the Rockefeller, and those biological discoveries now need to go to that next step to really make a difference to the benefit of mankind. And women are really poised now to be able to do that. So I'm going to turn over the podium at this point to Dr. Yejing Ge, and she is a postdoctoral fellow in my laboratory, currently deciding amongst the various different job offers for tenure-track university positions. But before she leaves, she's going to tell us about the various different ways in which she's moved her science now to the forward level of translation. She'll be followed by Samantha Larsen, who will then finish it off with telling us about her studies on psoriasis. So Yejing.
All right, thanks, Elaine. Thanks, everyone, for being here. It's really my true pleasure to be part of the conversation this morning, rise and shine. So For the next 10 minutes or so, what I'm going to do is tell you a little bit more, dive a little bit deeper into some of the questions that we have been passionate in the laboratory, and in particular how we are thinking about taking some of the discovery forward, as Elaine just suggested, to a level that we can start to think about, translate some of the finding in the clinics. So I will be focusing on this topic that cancer is a wound that never heals. I will tell you how it develops. So first of all, the disease I'm going to focus on today is human cancer. And as many of you already know, that it's really a complex disease at many different levels. You might have heard of some of the challenges we're facing these days. Cancer is not one disease. It's many, many different kind of diseases. And if you think about the current cancer therapies, one of the major problems that people are facing is a lot of these effective therapies, for example, chemo or radiation, you have a very strong collateral damage in the human patients. You might not think of that way, but these human patients are really devastated because their normal tissue are also hurted by the drugs or the chemo that's hitting on them while blocking the cancer. And there's this issue of therapeutic resistance. Even with the most effective ways, let's say the immune therapy that's really revolutionized our treatments, a lot of times the disease are not effective in a large cohort of patients. And even more daunting is more often than not, the disease actually come back. So it's effective at the beginning, but then they come back. And largely it's could be driven by the stem cell that Elaine had told you about. So I'm gonna use this analogy because it really speaks to me. I feel like right now, the way that we are looking at cancer feels like these blind mice with an elephant, that we are really capturing a tiny little bit from each different angles. And by doing that, we're never gonna figure out the broad picture, the big picture of really cancer, what cancer is and how we're gonna treat it. So how about, I'm gonna propose today to you a different way of thinking, to think of cancer differently and see if I can be, I like to be convincing that point. So there's this age-old hypothesis, but before I tell you the hypothesis, I'll give you a few glimpses of the historical perspective, people and human approaching cancer. So the oldest record in human kind of history of recording cancer dates back to the ancient Egypt. So I'm showing a picture of it's a surgery trauma book. And there, it was the first documentation ever in the history that there are 8 tumors that are being exercised by the physician here using the tool called fire drills by cauterization. I would view this is perhaps the most premature or primarily cancer therapy if you think about it. Now, fast forward to the very close by to us, but still a long time ago, is this German pathologist, this gentleman, Rudolf Virchow. He was the first one that ever showed a microscopic picture of how the cancer looked like, right? He's the founding, we would say that he's a founding father of pathology. But also what he proposed at the time when he was looking at these cancers, that this hypothesis, cancer is caused by chronic severe irritations. Of course, we now know that cancer is caused by genetic mutation and many, many different kinds. But the idea that a cancer and wounds might have a very close association really inspired a lot of biologists to go forward to ask this question and think about how to treat cancer that way. So it's actually not so uncommon. You might have heard it quite a bit that human patients with chronic wounds are largely predisposed to cancer development in many different types of tissues. And now, if you take a biopsy in the wound bed or take a biopsy from the tumor and compare and ask what gene that they express, they actually have a very strikingly similar expression pattern. The protein that they have are very similar. So why is that? And most importantly, if we know that phenotypic resemblance exists, can we learn something about this connection and try to think of cancer and treat them differently? So I'm going to return to this topic, one of our favorites in the lab. Under Elaine's tutorage, I am a stem cell biologist in training, so I think of human disease a lot from this perspective. So you've heard, Elaine, that we have in our tissues with these adult stem cells that really maintain or rejuvenate and regenerate the tissue in the daily business. And what's important also is upon stress. So these are cells that if you think about it, the first point to take home is that they are essential, because when an individual encounter an injury or stress, you need to rely on those cells in order to repair or to mediate the wound healing process. But if you think about it, this design of natural evolution actually really predispose some individuals, especially those under chronic wound or chronic stress, that these cells actually can get malignant transformation and become the fuel of the cancer. So what are we going to do about it? And if that is, I'm going to give you the punchline right here and show some data very quickly, but that cancer is actually fueled by transformed and mutated, the stem cell that goes awry. And those are the stem cells that normally would mediate wound repair in your tissue, but that they somehow cannot stop in cancer. And that's where we are trying to tackle. So I'll give you one slide of a quick flavor how the data look like and what that led us to think about this question this way. So this is a cross-section of the back skin of a laboratory mouse. And I'm highlighting here or showing here in two different colors, and they are marking two different lineage of the skin. One is the epidermis, and the other is a hair follicle. So you'll see that these are compartmentalized. They don't really overlap with each other. However, when you make a wound or the mouse inflict a wound in the skin, what you right away will see that these originally green cells start to express this red marker. So they have this mixed lineage gene expression pattern during the wound repair. However, the wounds heal. So in the mouse, the wound that we make, it takes about 2 weeks. So when the wounds heal, take a look at the skin again, you'll see that these compartments now separated. So they resolve, and then they become normal homeostatic pattern. Now what's striking is in cancer. So if you look at a section of the cancer, what you often find more is these cells that always become double positive for the two different lineage markers. And this never occurs in the normal skin, and this is only transient in the wound repair. So we found that we use this name stem cell lineage infidelity to describe this phenotype. And effectively, what happens here is you have the mixed lineage expression transiently during wound repair, but this phenotype gets sustained in cancer. This is just one quick example, and we really did a lot of work in trying to understand what is driving it, what's the root of this phenotype. But the last slide I want to show you is really to think about, we found this phenotype, and perhaps we can think about some ways to find a better cancer treatment. So here, I'm highlighting one of many observations that we had in the current story. So I'm marking, here is a cross-section of a tumor again, but now here I'm marking in red a gene regulatory element that driving a reporter expression in the tumor. So basically, these are the signal that highlighting the cancer stem cells or the cell that drive cancer development in a particular tumor. And you'll see also next to it is a wound bed. So some of the cells are also highlighted in green in the wound. But importantly, this signal is never seen in the normal skin in either lineage or any of there. So you can now think about perhaps we can focus on some of those signatures that selectively highlight the cancer stem cells, but not in the normal, and think about the strategy to kill them in this selective way. So the idea that we are trying to push forward with the Robertson Therapeutic Fund and to even moving forward is to really zoom on into the signature, which is selective to the cancer, but never in the normal, so that we can think of therapeutics that will not hurt the normal tissue, which is the problem of a lot of drugs these days. So a summary of what I just told you, that is we really started from this age-old hypothesis that cancer is a wound that never heals. And we showed that the cancer is really filled by stem cells, normally driving wound repair, but wasn't able to stop. And this phenotype is functionally relevant in both stress responses, I would say. But you can think about a selective cohort of genes or pathway to really kill cancer without hurting the normal. So with that, I'll turn the podium to Sam, our graduate fellow in the lab, who will tell you more of an interesting story in the other disease. Thanks.
So, good morning. My name is Samantha Larsen, and I'm a graduate fellow in Elaine's lab. And I would like to share, well, first I'll thank you for the wonderful opportunity for me to have the opportunity to share with you some of the work that I've been doing in collaboration with Dr. Shruti Nayak. And we're focusing on how can we target stem cells for the treatment of psoriasis. So first, a little bit about psoriasis. Psoriasis is a chronic, debilitating disease that affects approximately 2% of Americans, and it costs us billions of dollars in terms of healthcare costs every single year. And what psoriasis is it's characterized by these hardened plaques and is due to an aberrant immune response that takes the skin from relatively a thin layer, and it causes it to hyperthicken. So you result in this huge plaque that's often incredibly uncomfortable and causes the patients a tremendous amount of discomfort and pain. And currently, there are very few therapies that are available for the treatment of psoriasis. In fact, the therapies that are available are either systemic, and they're aimed at targeting those aberrant immune responses, and they try to dampen them through blocking certain parts of the immune system. However, a lot of these alarm signals that are associated with psoriasis are really important during normal body systems, and therefore blocking them actually causes really serious deleterious side effects. Then we have different topical treatments that are available, such as steroids. But if we constantly treat with steroids, what ends up happening is we dampen the immune system to such an extent that these patients actually become susceptible to other types of infections. And lastly, UV radiation. So these patients go to the clinic and will actually be exposed to short-wave and long-wave UV radiation, but what we know about sun exposure is that prolonged exposure to the sun actually in and of itself can cause and elevate your risk of different types of skin cancer. So what this really highlights is a significant unmet need for new efficacious therapy. And more important than that, starting to look at the problem of psoriasis from a different angle. So as Elaine alluded to, Shruti and I entered this problem looking at it from a very different angle. We were really interested in understanding why does psoriasis constantly relapse in the exact same place. So what we decided to do was model this in a mouse. And what we were able to do is selectively cause inflammation, like a psoriatic plaque, in part of the back of a mouse, and show that the pathology looks very similar to what we saw in humans. That we have in the control side or untreated side a epidermis that looks very normal, and then on the treated side where there's inflammation, there's now hyperthickening, and now the skin is a lot thicker than what it was before. So now, again, asking why does psoriasis remit and relapse in the exact same place, and making use of what we know about skin stem cells, these stem cells reside within the basal layer, or the bottom layer, the deepest layer of the skin, and they last long term. So we wanted to understand what were the long-term consequences of inflammation on these stem cells. So what we did was we decided to look, and what we found was that these stem cells play an active role in inflammation. They expand in response to inflammation, and they contribute to this hyperthickening and expand to this thickened epidermis. But most importantly, these stem cells survive that inflammation, and they continue to reside in this basal layer. So in a mouse, we looked up to 6 months after we caused inflammation, and we found that they continue to live on. And this is the equivalent to 15 to 20 years in humans. So now this makes it a really exciting and tantalizing place and a point of intervention, because if these are long-lived, could they potentially be some sort of harborer of information about the inflammation that they experienced that can prevent a new therapeutic intervention? So Shruti and I isolated these stem cells from the skin and characterized them. And our work was recently published as an article in Nature magazine. And what we identified was that these stem cells retain a memory of inflammation that enables them to respond faster to secondary challenges. So in the case of psoriasis, where it's remitting and relapsing, these stem cells are already primed because they've experienced a psoriatic flare before to respond and cause the psoriatic flare to occur again. So how we did this was we isolated DNA. So this memory is encoded at the level of DNA. And what we found was during an active inflammatory response, so during what would be a psoriatic flare, the DNA gets marked by these different markers that are indicative of inflammation. But what we found, which was really amazing, was that these marks are retained long-term, so they don't go away. So now that the stem cells have experienced this inflammation, they remember the fact that they have experienced it and have these marks. But these marks to the stem cells require different elements to come into play to result in a phenotype or to result in pathology. So these different marks need to be interpreted by the cells. So Shruti and I were really interested, can we mask these marks so that the stem cells don't recognize that they're there, and therefore we can block that psoriatic flare from reoccurring. So this is now one of the projects that's being funded by the Robertson Therapeutic Development Fund, and it's aimed at identifying novel inhibitors to target stem cell memory in psoriasis. So one of the inhibitors that we've been working with is something called JQ1, and this inhibitor is already going through clinical trials for other types of diseases, such as different types of cancer. And what we're working with is trying to repurpose this inhibitor and make it fit our needs for treating psoriasis. Additionally, another project that I have funding for through the Robertson Therapeutic Development Fund, but won't have too much time to share with you today, but would be happy to answer questions later, is through our studies, we also recognized that there are certain transcription factors which are necessary for gene expression that are really critical for healthy wound healing. That when you get older, they start to go awry and they're in disarray. And what ends up happening is you result with chronic wounds or wounds that don't heal properly. So we're screening for activators of these transcription factors that can be employed to help return wound healing to what it should be in younger individuals. So now a little brief update going back to Project 1, where we are looking for novel inhibitors to block memory in psoriasis. I'm going to walk you through now, switching gears a little bit towards the entrepreneurial side and where we are in the business pipeline. So first, what we're working on is establishing novel intellectual property. So as I mentioned before, this is starting with a compound that is already going through clinical trials. And what we're working with is through our Robertson Therapeutic Development Fund, we've actually piqued the interest of an organization that's tri-institutional called the Therapeutic Discovery Institute. And this is available at Rockefeller as well as Weill Cornell and Sloan Kettering. And what they do is they pair basic scientists up with organic chemists as well as patent lawyers and people with technology with knowledge of the biotechnology sphere. And we create a team and we all work together to create a compound that can go through to a clinical trial. So what we are working with now is taking that compound and making it so that we would have novel intellectual property by first making it such that it could be applied topically. So as I mentioned, these psoriatic flares occur and remit in the same spots. So there shouldn't be a need to take a systemic drug because you want to target the specific area that you have this flare. So we want it to be treated topically. And secondly, we want it to break down as soon as it hits your bloodstream. Therefore, you won't have any systemic side effects. So working with these organic chemists across the street, we've already developed about 30 novel compounds that we are now have to demonstrate a proof of concept. So again, this therapeutic development initiative has actually paired us up with a company called Epistem. So what Epistem specializes in is working with psoriatic skin rafts in culture in a dish. So this company will now screen through these 30 different novel compounds for Shruti and I, and at the point at which we can prove its efficacy in a dish, we now get these compounds back and we can now test them in humanized mouse models. And through this entire process with Rockefeller as well as TDI, Shruti and I have been in constant communications with different patent lawyers, working to understand how do we protect our IP and how do we move forward ultimately to be able to file and move forward. And if you're wondering how, on top of getting my Ph.D. in science, how I'm trying to manage and keep up with all of this business information, which does at many times go over my head, there is a fantastic course that is available that Shruti, Yejing, and I all took called the Tri-Institutional Bench to Bedside Initiative. And this is a 3-month crash course on how to teach scientists to think like entrepreneurs. And what we do is we actually license technology from one of the 3 institutes and create a biotech startup. And this is a fantastic opportunity where we really get the ability to work with different patent lawyers, work with technology officers, and understand what it is to present in front of venture capitals and pharma so that we can start to have that experience at a much younger and earlier stage of our careers. So lastly, there's now this harmony that needs to occur, and it is starting to occur at Rockefeller between scientists and entrepreneurs. And what we're working on is how do we start earlier on to take what we discover at the bench and translate it into what could potentially be something that goes to the clinic or even start our own company. So hopefully the next time somebody is asked to draw what a leader might look like, it might look a little bit more something like this. All right, thank you. Perfect.
Thank you for these three extraordinary presentations. I'd now like to invite our other speakers up to the podium, and we have time to take some questions. If you have a question, please raise your hand. We have roving microphones that will come find you. Well, perhaps I'll start by asking Elaine how these different dimensions are affecting the culture of your laboratory.
Yeah, so I love the question because this initiative for getting involved in the Robertson Fund started with Shruti Nayak, who isn't here today, but Sam alluded to the research. And that was our first opportunity to really start thinking about how do we actually take our findings and make them go to the next step of really thinking about taking them to the clinical level. That then has developed into an incredible excitement and interest in the laboratory, where I think just last week, there's another woman, Sichi Liu, in my laboratory who received Robertson to look at another aspect of inflammation with a different avenue and different drugs. And now I think there are 5 or 6 people in my laboratory who have taken that initiative and gone to the next step. And so I think now the flavor of the laboratory is that we're constantly thinking about how is it that we can take our basic science findings and really bring them to the next step. And I think what is really needed now is this even earlier stage of funding, and as Samantha mentioned, the importance of training in mentorship, because we haven't traditionally been thinking about how we can do this. And I think now that it's on our radar screen, we're really excited about it. And I think when you start to magnify that with all the women heads of laboratories now at Rockefeller, it's going to be a very exciting time.
Great. Thank you. Oh, yes, microphone. Yeah.
Hi. How do you determine that the same work is not being done elsewhere?
That is, yeah, so how is the same work not being done elsewhere? The scientific community is pretty much on target with that. Everyone in my laboratory who is working on various different projects is always cognizant of the literature behind us. We can never be sure that there's not someone else who's working, who has the same idea that we do and is out there working on the research projects at the same time, but still haven't published it. That's always a bit of a concern, but I think we've gained the confidence over the years that if we have a really good idea and approach it in an unconventional way, that chances are there's nobody else out there that is going to be doing the exact same experiments that we are. And I would say that more often than not, that's borne itself out.
I think Elaine's being a little bit modest. You know, Satchel Paige once said, never look over your shoulder. You never know when somebody might be catching up. Elaine is so far ahead of the field that it's usually unlikely that anybody else is going to be doing the same things, at least certainly at the same level as Elaine. Yes.
I was wondering if the work you're doing and the work that Jim Kruger is doing, how far apart, and if there's any ability at this point that the two, the clinical and the lab, have practical uses at this moment.
Lovely question. So Jim Kruger is another one of our faculty members who actually works on the human side of psoriasis, and his approaches have been very, very different than ours. coming at it from very different angles, we're getting to the point where there's starting to be, that gap is starting to be closed. And I think that is, again, another interesting way in which science can meet science from different angles. And that's where I think real advances are going to be moved forward. And so Jim has talents in the clinical side that we don't have. We have talents in the molecular biology, coming up with new ideas side that maybe his laboratory doesn't have. And I think together, this could be very going in a very exciting direction for us in the future.
I can quickly add to Elaine's point because I have some personal experience with that. The laboratory model that we're using, that Elaine has developed, the lab has developed in the field, the idea is sometimes if you look at disease, right, you compare the disease and normal, you see signature, you see a very strong different gene programs that's manifesting in the disease but not in the normal. Now the question is, are these genes responsible for driving the disease? And many often, if it's not, it's just expressing there, it's really difficult to come in and then find a drug that will target these genes and cure the disease. So we have this power in the mouse laboratory that we can screen in a very high throughput fashion in vivo to find which gene that you see the change in the normal and disease, but also a driver of the disease. I think that's a power that we're using mouse as a model. And then the human side, as Elaine has alluded to, then we can go back and see, well, is that the signature you see in the real human patients? So that really, I would say that Elaine has said that really marry these two approaches very seamlessly together.
Question here.
To follow up on that and ask about the science, because when you show the injury, the wound healing injury to the mouse, you have two different types of stem cells, one that repairs the epidermis and one that repairs the hair follicle. And suddenly they're both doing both jobs? And is that because each cell becomes capable in itself of doing both jobs, or is it because the cells are fusing in some way so that they're joining together to do it?
I love the
And
question.
why would they be
Yeah.
separate in the first place if
Yeah.
they always have to do
Yeah.
both?
So we've been working on that very question in a broader sense of the laboratory. There are several different people in my lab working on wound repair and trying to understand how to optimize it. And what we've learned is that it's almost like a 911 call in the skin. The stem cells of the hair follicle, the stem cells of the epidermis, the stem cells even of sweat glands. In a 911 call, if there's an injury over here, it's the closest stem cells to the scene of the accident that come into play and that become mobilized. We call it stem cell plasticity. Yejing, in her discussion of stem cells and wounding and cancer, we've referred to that as lineage infidelity. But it's this plasticity that stem cells have, which I think is really fascinating, that allows a hair follicle stem cell, if there's a wound over here, to be mobilized, come up, repair it, and thereafter it will behave like an epidermal stem cell. And so we've been learning about the cues and about the plasticity. We can't take an epidermal stem cell without genetically manipulating it and getting it to make a neuron. But we can take the natural system is that the stem cells will have this plasticity. And again, I think it's an evolutionary advantage to be able to repair wounds as quickly as possible.
Right behind, yeah. And then Lulu.
There has been discussion about psoriasis, and you talk about looking at mice. And my question was, how do you cause a mouse to get psoriasis so that you could do your experimentation?
Sam.
That's a great question. So there are a couple of mouse models that are available to us to mimic what we would see in psoriasis, and there are no perfect models. The one that Shruti and I focused on is called a Mickle mode. And it was actually, believe it or not, used to treat atopic dermatitis back in the day, but in mice causes psoriasis. Different receptors. But we can also inject the different pro-inflammatory alarmins that cause psoriasis. We know that they cause psoriasis in humans, and if we inject the mice with those same alarmins, it recapitulates the pathology, and we can cause the same formation of psoriatic plaques.
Elaine, you start off early on by saying that as we age, our stem cells are less active, less able to do their important work. And given that they have such an important role in our overall health and wound healing, what kind of work is being done now to try to prolong the robust life of our stem cells?
Yes. So we looked at this question, Lulu, from the perspective of the hair follicle, and we figured out what signals are the go signals and the stop signals for these stem cells. What prompts them to make hair? What prompts them to stop making hair? So we thought we were there at the fountain of youth, where all we had to do was basically stimulate those hair follicles that had the stop signs at an elevated level, and we would be there. If we just turned on the go signals, we'd be able to rejuvenate the hair follicles. What happened is that we did that. And so at first, it looked great. And in fact, there are several different companies that have picked up, before we've gotten our act together through entrepreneurship, that have picked up on our findings and are actually marketing, working to market those drugs that will stimulate hair growth. We're a little bit of one step ahead of them, though, and it's in an unfortunate way. What we've realized, when we constitutively cause mice to crank out hair, and they can do it nonstop, they run out of gas. The stem cells run out of gas. So now we're back at the drawing board, because what we've realized is that the stem cells, which typically are only utilized for when they need to be, and really predominantly for repairing wounds rather than, unfortunately, replenishing our hair follicles, what we're beginning to realize is that those stem cells appear to have a limited lifespan. So now we're back at the drawing board, really trying to study the lifespan of a stem cell, and then can we do something about that to extend their lifespan? So I think it's going to be an interesting road. Wasn't quite as simple as one of my other graduate students thought it might be when he saw dollar signs in front of his eyes at night. But I think it's a fascinating question.
Thank you. I think the hour is late, and we'll have to draw to a close. For those of you who have more questions, I'm sure Elaine and Yejing and Samantha will be willing to stick around and answer a few more questions. But I'd like to thank all of you for coming today, and particularly thank the speakers for such a stimulating presentation and for your questions. Thank you so much. Look forward to seeing you again on campus.